What beverage? Okay, well, it's my pleasure, everybody, to introduce Stepfanie Aguillon to our group. I first met Stephanie, I think it was like 12 or 13 years ago, when she was maybe an undergrad or a master's student giving a poster on bluebirds at an ornithology conference. And it was a really interesting poster, and it's been fun following her career ever since. It's been over a decade since then. And now she's a new faculty member right up the road from us at UCLA. She just started there a couple of years ago. So it's exciting to see everything she's done. I'm excited to have her come talk to, or not come, but talk to us tonight. Her work focuses on how new species are formed, and she combines fieldwork, museum specimens, and genomics to learn how biodiversity itself is generated. And that's a pretty important subject. I mean, we all care about biodiversity, and part of learning about biodiversity and knowing how to conserve biodiversity is understanding how it's generated in the first place. And so today she's going to talk specifically about her work with flickers. She's worked with a variety of species, and the flickers, I guess, is going to give her an opportunity to present some of her really interesting, intriguing research on color. So I think we're going to hear about color too, I hope. Stephanie's originally from Arizona and got her BS and MS from the University of Arizona in the Department of Ecology and Evolutionary Biology. And then after that, well, she went to Cornell University, which we know is Bird Central, and got her PhD there in the same field in 2021. And after getting her PhD, she went to Stanford for a postdoctoral fellowship for a few years before becoming a professor at UCLA. And she's also a research associate at the Los Angeles Museum of Natural History. So she has that association as well. Her work has already been recognized with early career awards from the American Ornithological Society and from the American Society of Naturalists. In addition, she's also an elected member of the American Ornithological Society, which is an honorary category. And beyond all this, she's just a very super nice person and always gives insightful talks and entertaining talks. So I'm really happy to have Stepfanie here with us tonight. So join me in welcoming Stepfanie.
Thank you so much. What a nice introduction. Okay, I actually don't remember meeting at that must have been AOS in Chicago in 2013, but yeah, that's a long time ago now. Um, okay, let me go ahead and share my screen so you can see. Here we go. Okay, you should see just my slides. I'm going to move my windows around here a little bit so that I can see slides and you all. Okay, so I'm going to tell you a little bit about my work on flickers tonight. I'll do a little bit, I've heard you do both identification as well as science talks. I'm going to sort of meld both of those together, do a little bit of bird ID and science, because I feel like that works really well with flickers. But I really like to start these talks with a bit about my academic journey. So Kevin introed this a little bit already, but just to give you an idea of, where I'm coming from and how I got to be where I am. So I grew up in Sierra Vista, Arizona, which is a place that to non-birders, they have no idea where this is. I will mention I'm from, southern Arizona or something. But for birders, if I tell you I grew up, my parents still live, basically right in Ramsey Canyon. I can get there in about 5 minutes in the car. So this is where I grew up. Grew up right on the border, didn't really know anything about birds. But I spent a lot of time in the Sonoran Desert, which is really, a biologically unique place. So lots of sunrises and beautiful sunsets, obviously. But a lot of really interesting organisms. So a lot of birds that you can't really see anywhere else in the US, many mammals as well you can't really see anywhere else, lots of herps. And I grew up spending a lot of time outdoors. So I spent time camping and fishing and hiking for much of my childhood. Here's some of that. And that really got me interested in biology and trying to understand the natural world. And I'm the first one in my family to go to college, but I, didn't go far. I went to the University of Arizona, and I was really interested in biology, and that's sort of led me to where I am today. And I studied bluebirds as an undergrad and as a master's student, which I guess is where Kevin first met me, talking about those bluebirds. And from there, I got really interested in birds after taking an ornithology class, actually. I was like, man, I could study birds forever. And, here I am still studying birds. So I think it was a pretty important class and important part of my journey here. And from there, I went on to Cornell, where I studied flickers, which I still work on today. I learned a lot about birds and a lot about how to do science there. I also learned a little bit about living in a different climate. So this is just some photos of some snowstorms during my time there. That is my car in that pile of snow that I had to shovel out because it needed to move to the other side of the street. Um, and learned a lot about birds, learned a lot about genomics and hybridization, which I'll tell you a little bit about today as well. And I also got the really special opportunity to travel to a lot of places because I decided to go to grad school. So Glacier National Park. I went to the Galapagos twice to teach a course. I got to see things like vegetarian finches and flightless cormorants. I got to go to Brazil and the Natural History Museum in London and many, many amazing places, all because I sort of took this really interesting path. And from Cornell, I went on to come to California. I went to Stanford and I took a few years to study fish, studying similar questions to what I've studied in birds, but working on them with organisms we could easily keep in the lab. And I think as many people who come to California realize, you get to California and you're like, man, I could just stay in California forever. And so I'm really lucky to have moved to UCLA and started my lab here. So I started here and have been in LA for just about a year and a half. Um, my lab really just opened, and we're mostly working on birds here, of a variety of types. I'll tell you a little bit about the work we've been doing on flickers. But much of what we focus on is coloration, and color's something that has really fascinated, especially ornithologists, but biologists generally, for a really long time. And one of the reasons for that, in ornithology in particular, is that bird specimens often maintain their color really, really nicely if they're preserved correctly. So these are a bunch of birds of a variety of colors here to show you the rainbow. And this interest in color has been long-standing in biology. So we can think about Darwin on the beagle, for instance. So he was really interested in color and describing color. This was done historically using this what's called Werner's Nomenclature of Color. This is a book that had color swatches, because you can imagine if you were in a time when there was no internet, you could not say, this is the color, let me send it to you. You had to say, it's this particular blue from this book. And so I think a lot about color, both in my professional life and my personal life. I do a lot of handcrafts as well. And then my research also focuses on hybridization. And I'll tell you how these two things intersect with each other. But hybridization, just to give you a bit of a definition, one example you might be familiar with is breeding a horse and a donkey. When you do this, you get a mule. You might know that mules are really hardy pack animals, but that they can't produce any offspring. So like, yay, pack animals if you're trying to go down to the Grand Canyon, and then sad if you're trying to produce any offspring from mules. And so you might think about this, and if this is the only example of hybridization I wouldn't be surprised. That's totally okay. But not all hybridization events are like this, actually. So, but I would forgive you if you thought this, because if you search hybrid animal, for instance, you might get organisms that look like this, which obviously are not real. But this is what, if you search, you're like, hybrid animal, great. But not all hybridization events in nature are actually evolutionary dead ends. So there's many examples, and we know this as we're sequencing more genomes across organisms. Many organisms have had hybridization in their past, things from yeast to flowers to butterflies to mammals. This includes humans. So humans have a history of hybridization with other ancient hominins. And this is really important for evolutionary trajectories today. And in birds in particular, as you might know if you're used to looking at birds in the wild, hybridization is really common in birds. So we have lots of examples of individuals of species that we know hybridize really regularly. These are just a handful of examples here. And one of the things I'll highlight to sort of dig into why I think about hybridization and color together is this warbler. You might know this warbler. This is a result of hybridization between a blue-winged and a golden-winged warbler. It produces what's known as a Lawrence's warbler. When these Lawrence's warblers were originally identified, they were thought to be a separate species. It was later realized that these are hybrids. One of the reasons we know that these are hybrids are based on the way they look. So this Lawrence's warbler gets its overall yellow coloration from the blue-winged parent, but it has this black eye mask, black bib from the golden-winged parent. And hopefully it's not too much of a stretch to say that this is the result of underlying genetic differences. So genes produce these different colors, and these are passed down to the hybrid individual. Now, I don't study warblers, so there is a ton of really interesting and exciting work on hybridization in warblers. I work on the northern flicker, which you may and likely know are two woodpeckers that are distributed across North America. Red-shafted flickers here in the west and sort of west of the Rocky Mountains, yellow-shafted flickers broadly in the east. And in the Great Plains, sort of roughly from northern Texas, following the Rockies up into Alaska, these birds hybridize and produce hybrid intermediate individuals. And because flickers have these great coloration differences, this is a really nice opportunity to also talk about identifying flickers. So I'm going to do a brief interlude before I talk about science here to talk about IDing plumage in flickers. So unlike other woodpeckers, you may know that flickers are commonly seen on the ground. I'll show you this brief video here of what you're likely to see if you're seeing a flicker. Let's see. Well, it's playing, but not playing. Let's refresh. And so you're likely to see, an individual hopping around on the ground. That's because flickers eat ants, and so they're commonly found in lawns, walking around, digging their face into the ground. If you get a close look, either in the hand or in binoculars or scope of a flicker, you'll actually often notice that their bills are covered in dry dirt because they spend a lot of their time with their face in the ground. So this is sort of where you would normally see a flicker. You may also commonly see them flying away as you flushed them, and you'll see their bright white rump. Okay, so that's where you'll commonly see them. You may also, of course, see them in cavities or in boxes. So flickers are, as are other woodpeckers, cavity nesters. These dig out cavities, both for roosting but also for nesting. So you may see them there. Flickers are also known for interacting with each other. So you may see them fighting either up in the trees. You may also see them fighting on the ground. So I wanted to show that video as well. So doing these stereotyped behavioral interactions. This is called the Wicca dance. So often with these interactions with each other. So these are two females, but males do this with each other. Females do this with each other. You may see two individuals, you may see multiple individuals. They often do this stereotyped sound, which is why this is called the Wicca dance. So you might see them dancing like this. Back to the slides here. But one of the things that I think is really interesting about these birds is that they differ in color across a number of plumage patches. So I think very obviously red-shafted and yellow-shafted flickers, you probably know they differ in the color in the wing and the tail, but they also differ in a number of traits in the head and the face as well. And I'll just put these up here right now. The malar stripe, the throat color, ear coverts, crown, nuchal patch. I'm going to walk through these with photos just to help with some identification. But the thing I'll say with these illustrations first is that, so yellow-shafted and red-shafted flickers have different colors here, but hybrids can have a variety of combinations of these traits. They can have mostly red-shafted traits with a few yellow-shafted in there. They can have intermediate colors, so you might have orange wing and tail the mallard stripe is red versus black. You might have sort of half red, half black, and all combinations in between. And I'll tell you why this is important for the science that I do towards the end. But just to say, hybrids can have a lot of interesting combinations. So one of the things that I think is important to talk to, especially with folks that are interested in looking at birds, is how we know, if we have a red-shafted or a yellow-shafted or a hybrid individual. You might also call hybrids intergrades. You can use those interchangeably. So either works. So let's start with the color of the wing and the tail. So red-shafted flickers will be on the left here on the slides. Yellow-shafted flickers will be on the right. So red-shafted flickers, as you might imagine, have this salmon pink color wings and tail. Yellow-shafted flickers have these really vibrant yellow. So you can see this if you're seeing the underside of the bird. You can also see this if you're seeing the top side of the bird, because the coloration is also there on the rachis of the feather. So you can see that on both the wing feathers and the tail feathers. So color of the wing and tail, I feel like, is a self-explanatory one, so I won't linger on this, but let's dig into some of the traits on the head and the face. So throat color and ear coverts. So I'm showing here arrows at the throat on the bottom and the ear coverts, which are the feathers covering where the ear would be if you could see the hole. Trust me that there is a hole there where the ear is. So in red-shafted flickers, both of these traits are gray. So you have gray ear coverts and gray throat, which is different to the yellow-shafted, which, showing you zoomed in yellow-shafted here, is tan for both the ear coverts and the throat. So we have gray and red-shafted, tan and yellow-shafted. And this is, actually quite easy to see if you have a scope or binoculars. And so it's not something where it's like, you'll never see this unless you have the bird in the hand. It's actually, once you get the hang of the actual colors that these should be, it's very easy to see when something looks off. Okay, and we can use these same pictures actually for the mallard stripe. So the mallard stripe is this region on the face that is either red or black. These are males, so this is visible in males. Red-shafted flickers have red, yellow-shafted flickers have black. It's this very broad stripe on the face. And this is the only trait that is helps you identify sex in these birds. So these are males. Males have red versus black. Females have no clearly visible mallard stripe. Some individuals that are red-shafted, you might see that it's a tan patch, but it's not bright red or bright black like it is in males. This is the only trait really that differentiates these birds by plumage for males versus females. And the birds actually care about this trait. So there were studies in the early 1900s where people captured females and colored their mallard stripe black, and their mates, their male mates, then treated them as males and tried to kick them off territories. So we know that the birds know this trait is important too. So, okay, we have ear coverts, throat, mallard stripe. We can also think about the coloration of the crown. So I've zoomed in here on different photos here. So red shafted, the crown here is brown. This is sort of color going from the bill sort of back to the nape of the neck. Yellow shafted have very clearly gray. And you can see how this is differentiated between crown versus sort of the lower part of the face. So the ear coverts and the throat are an opposite color from the crown, and that's true in red-shafted and yellow-shafted. It's just different colors. They've swapped. And this is a trait that I will say has a little bit of variation, especially in red-shafted flickers. So you might see individuals that have really dark, rusty crowns that are really clearly distinguishable from the ear coverts and the throat. You might see individuals that have not that really strong differentiation, where most of the rusty color is sort of towards the bill and up to the eye, but doesn't really extend back. So that's pretty normal variation in red-shafted flickers. Yellow-shafted flickers don't have this amount of variation, but just to say red-shafted flickers tend to be variable in this way. And then the other trait that is pretty interesting is the nuchal patch. So this is the patch on the nape of the neck. So I'll have birds in a different orientation here. So in red-shafted flickers, this is completely absent. So there is no bright red patch on the nape of the neck, which is really different from yellow-shafted flickers, which have this really bright, really broad red patch. So this one is a presence versus absence. And this is both in males and females. So males and females both have this broad patch or don't have it, depending on red-shafted versus yellow-shafted. I almost for yellow shafted put this image, which maybe will help you remember a little bit. Some yellow shafted females fighting with each other and grabbing the nuchal patch here on each other. So yellow shafted flickers have this nuchal patch. And this is another trait. So I talked about the crown in red shafted flickers, but this is another trait that there is some variation in red shafted flickers. So yellow shafted flickers always have this really broad red patch on the nape of the neck. Red-shafted flickers have a bit of variation. You might see individuals with a tiny bit of red in the nape. You might see individuals with quite a lot of red in the nape. And this is particularly common in the Pacific Northwest. So in Washington, in Oregon, you see birds that look like this. We don't know if this means these birds are hybrids. This is something I'm really interested in trying to know here in the future. But it does seem like this nuchal patch trait is one where there is a bit of variation in a way that is not just like always absent. And so these birds in particular, if all of the other traits look like red-shafted flickers, I would tend to call these red-shafted flickers rather than intergrades. Especially if it's just the nape of the neck that has a little bit of red, but everything else looks red-shafted. I would probably call those red-shafted rather than intergrades. So, okay, let's sum up here. Talked about broad differences in red-shafted versus yellow-shafted here. I'll mostly show these illustrations moving forward. But hopefully, this is to say if you see a bird either on the ground or in a tree, you should be able to pretty easily identify red-shafted versus yellow-shafted based on traits on the face, based on the wing and tail. And so I thought what I would do is show you here a few images. And often you'll have a sense that birds, you might expect hybrids based on particular places. If you are near the hybrid zone, sort of going from following the Rockies up from northern Texas up to Alaska, you might see birds that are not pure red-shafted and not pure yellow-shafted. So I'm showing a few images here. I'm going to give like a few seconds here for you all to look at these and to try and see if you can clock the things that make these birds interesting and why I would call these hybrids or intergrades. These birds on the bottom actually are birds sighted in the same yard, which I thought was fun. And all the photos I've shown so far are from eBird, from Macaulay Library. So these are all sightings from folks like you. Okay, so thinking about this bird in Calgary, one of the things when I see this that really rings alarm bells for me is the face is tan and the crown is brown, but all of the other traits are red-shafted like. So this bird has these yellow-shafted ear coverts and throat, but all other red-shafted traits as far as I can tell. So I would call this one a hybrid or an intergrade. This bird in Washington has red-shafted like traits on the head and the face, except for its malar stripe is black, which is pretty interesting and different. So I would also call this guy an intergrade. This bird from Colorado, you might notice it has a nuchal patch and a red malar stripe. So it has sort of red-shafted like traits, but it also has a nuchal patch. But the ear coverts also are a bit intermediate. They're not really tan, they're not really gray, they're sort of in the middle. And then one of the sort of bigger things, you might notice that you can see the color of the wing and the tail just based off of the rachis of the feathers. And the wings have yellow, but the tail have red. So this individual has a lot of interesting things going on potentially. And then this bird in Washington, this last one, seems yellow shafted, at least from the wing and the tail, but has a red malar stripe. And then just nuchal patch maybe looks a little small, though it's hard to tell exactly from this photo. So hopefully these felt like you could look at these and say like, oh, there's something off here. This doesn't look exactly like a yellow shafted or doesn't look exactly like a red shafted. Okay, so that's a little bit of identification, but I thought now I will tell you about two science stories using coloration here in these birds. The first one is about plumage color in the hybrid zone, and the second is thinking about genetics and thinking about the genetic basis of these color differences. So let's start with this first one here. So there's actually a long history of study of the flicker hybrid zone. So again, just to remind you geographically, this hybrid zone is here shown in orange, sort of in the middle of the continent in the Great Plains and following the Rockies. There's a little bit, so this sort of bends towards Washington and the Pacific Northwest. And so there is a bit more hybrids there, but that's not really ever been studied very intensely. But what has been done is work in Colorado and Nebraska, and some intense collections in the 1950s along the Platte River, which is this river that goes east-west across the Great Plains. And we know that as you go from west to east, you grade from red shafted-like traits through to yellow shafted-like traits. And this is thinking about all of these color traits that I just told you about. If you sort of summarize these in a score, you start from red shafted over to yellow shafted in the east. And the great thing about this collection is that these birds are housed in the museum where I did my PhD, the Cornell University Museum of Vertebrates. So we use these for events of the public, like what's shown here. But this also was a really great opportunity to try to resample this hybrid zone, to do repeated sampling, which is one of the things that I did during my PhD. So we went back out to the Platte River and sampled the original locations and others where these birds were sampled to try to understand how the hybrid zone has changed in 60 years. And so that's the question that we'll be answering here in this section is what's happened to the hybrid zone in the last 60 years? So we've conducted this repeat sampling. So in the historic sampling in the 1950s, we had about 250 birds. And in the contemporary sampling, we had a little over 100. And this was all work that was conducted with Vanya Rower, who's the curator of birds and mammals at the CUMV. So shout out to Vanya here. And what we're able to do from this collection and from our knowledge about plumage color here is to create what's called a hybrid index, which is basically summarizing all of those coloration differences I told you about in a score that ranges from 0 to 1. So if you have all yellow shafted traits and they look exactly as you would expect in a yellow shafted flicker, you get a score of 0. If you have all red shafted traits that look exactly as they would in a red shafted individual, you get a score of 1. And then you're somewhere in between otherwise. And just to remind you, we talked about these traits already, wing and the tail, traits on the head and the face. We have a score for each of these, and then we add this up and average it to get and standardize it to get a score from 0 to 1. And then we create what's called geographic clines. I'm going to walk you through how to read a cline. So clines look something like this, where we have this geographic cline. And on the y-axis, we have phenotypic traits. So say the color of the wing and the tail, and distance along the x-axis. So with these traits, we have values for species 1 on one side, sort of up at the top of this phenotypic trait. And then values for the other species on the other end, sort of at opposite sides. As you're in the hybrid zone, you see quickly go from one species trait to the other. These are individuals that are intermediate in plumage. They give you this really sharp transition from one species trait to the other. And when we do this geographic cline analysis in flickers, what we see is that the hybrid zone has shifted about 73 kilometers west. So we can create this cline for the historic sampling. We get something that looks like this. Again, we grade from red shafted-like traits over to yellow shafted-like traits. And when we do this for the contemporary sampling, we see that this has moved significantly to the left on this plot, which is west. And this is a summary of all traits, but we see this as well if we pull out these traits individually. So we see across the overall hybrid index, this movement, but across a number of the individual traits as well. In particular, the wing and the tail color, the nuchal patch, the crown. And if we look at what this looks like on the map, it's something like this. So we have movement from sort of the corner of Nebraska and Colorado in the 1950s. And then this has shifted significantly towards the Rocky Mountains in the 2010s, much more into Colorado. And this movement has likely occurred rapidly in the last 30 to 40 years. So from previous studies of this hybrid zone in the early 1980s, we know that this was stable. There was no movement up to the 1980s. So this movement must have occurred since then. And so this is maybe the result of climate change. We know from other studies of hybrid zones that they have moved because of climate change, and this is something that has been able to be documented. In flickers, we don't exactly know if there are particular climatic variables that might be influencing this movement. This is something that I'm hoping folks in my lab will be able to answer. It's also possible that this is the result of land management changes. So as you might expect, we're planting more big trees as places have become colonized, and this is true in the Great Plains. So there's been increased planting of trees, which has really changed the way the Platte River flows. And this potentially has changed, movement of red-shafted and yellow-shafted flickers. So you can sort of imagine if you're in the Great Plains, one side is the Rocky Mountains, one side is just eastern forests. If there are more trees now in the Great Plains, it's potentially easier if you're a yellow-shafted flicker to move into that river valley than it is if you're a red-shafted flicker and you're already having to go over the Rocky Mountains. Potentially, it's easier if you're yellow-shafted to just come in, which could result in this movement west. We don't really know. These are things we're interested in looking into. But thinking about sort of this broad question, how has the hybrid zone changed over the last 60 years? So using just plumage, we know that this hybrid zone has moved significantly. So doing a similar thing to what I outlined for you at the beginning, thinking about if these traits are tan versus gray, red versus yellow, black versus red, we can see that the hybrid zone has moved over the past 60 years. It's shifted significantly west. We can see this in the declines. We can see this when we look at where the hybrid zone is centered on the map. And we're trying to figure out why that is, but still TBD. And then, so this is thinking about just plumage for this first story. But now I'll tell you a little bit about the genetics, which are actually really interesting in flickers. So hybrid zones are really great opportunities for thinking about the genetic basis of color. So as I mentioned, flickers hybrids have these interesting mix and match of traits. So they have different combinations of colors. They have intermediate colors. You might have an individual that's completely yellow shafted except for, the malar stripe is red, or any combination really. And this makes hybrids really interesting. And in particular, because this mix and match is also true in the genome. So if you imagine the genome as a line with different colors, the different colors indicate where that piece of the genome came from, yellow for yellow shafted, red for red shafted. And we can use this mix and match to try to identify where in the genome creates a particular color. So say we're interested in the color of the wing and the tail for red shafted individuals. We want to know where this red comes from. We can look in these genomes and see which region those individuals share and that are not shared with individuals that don't have red. So in this sort of toy example, we have this region at the top of this genome that's red and these red shafted individuals that's shared between those two, but not in the other individuals. And so this is a toy example, but we can do this with real genomes, with real individuals. And this is one of the things that I've been really interested in and have been thinking about for a long time. So let's think about the genetic basis of color in flickers. What is creating these coloration differences? So to answer a question like this, I'll tell you a little bit about the method. So this involves some kind of DNA sample. This is either a blood sample or a tissue sample. I've done work both where we're collecting for museums as well as work where we're catching individuals and releasing them. What you need is some kind of DNA. This ends up being a lot of molecular lab work. So I got interested in this career in science for, interest in being outside in nature. I now spend a lot of time doing both molecular lab work, but also computer work. So I would say I spend much more time on these latter two. And this produces, in this case, 7.25 million markers. And I'll tell you what I mean by markers. So genomic markers of differentiation is what we're getting here. And let's think about DNA. So if we have three individuals, we can see their DNA. We have one individual that's red shafted, two that are yellow shafted. We can look across these base pairs and see which ones are the same and which ones are different. In this case, I'm showing you this one base pair right here in red that's different. Everything else is the same. So the two yellow shafted individuals have a C here, the red shafted individual has a G, and this is a marker of differentiation. You might hear this called a SNP, a single nucleotide polymorphism. This is a region of the genome that differs and can tell us a lot about differentiation. So we have 7.25 million of these markers across the genome in flickers, and we can use these to look at where differences are between these birds. So let's look at a single chromosome first, and then I'll show you the whole genome. So these plots are called Manhattan plots, and I'll walk you through how to read them. So the y-axis has this measure of differentiation called FST. This ranges from 0 to 1. All you really need to know about this is that 0 means a marker is completely identical, 1 means a marker is completely different. The y-axis or the x-axis here is position along the genome. This is showing you chromosome 10. So as you go from left to right, you're moving left to right along the chromosome. Hopefully you see as you go left to right, you have similar, 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 and then this region that's really different. These plots are called Manhattan plots because you can imagine looking for these skyscrapers amongst the city skyline. So you're looking for these regions that pop out as different. So looking across, this is just a single chromosome. We can do this type of analysis for the whole genome as well. And when we do this, we see really low differentiation, but a number of these skyscrapers. So this is what it looks like when you compare red shafted and yellow shafted flickers that are far away from the hybrid zone. So if you can imagine trying to plot 7.25 million points on this graph, you can imagine that would be really hard, and you can't really see them all here, obviously, but the average of all of these points is 0.008. So remember, if we're thinking if these birds are genetically similar or, completely different is 1. So FST of 1 is completely different. Completely identical is FST of 0. So if our average across the 0 is 0.008, these birds are nearly genetically identical. You can still see a number of these skyscrapers. So these are regions where these birds do have strong differences. So there are regions that are different, but these birds are actually really similar. And in this panel of 7.25 million markers, we have less than 1,000 that are completely different between red shafted and yellow shafted. And so these birds are birds that are far from the hybrid zone. These are birds from New York and Florida for yellow shafted and California and Oregon for red shafted. So even far away from the hybrid zone, these birds are nearly genetically identical. But remember the potential of hybrids. So if we look at these hybrids, we can try to do this association between genomic regions and the color. And when we do this, we actually find multiple regions of the genome associated with color. So thinking just about the wing and the tail, we can do this analysis looking for an association. This is a Manhattan plot. The only difference here is now the y-axis is the significance of a particular genomic region being associated with a color. So for the wing and the tail, we see a number of these regions that are really strongly associated with color. We can do this for these other traits as well, and we find associations there too. For the sake of this talk, I'll just highlight moving to the gene level. So looking within these regions where we see this strong association, we find a number of genes that are related to pigmentation, some related to melanins. So melanins produce the blacks and the browns and the grays that we see in nature. There's a number of genes related to melanins. We also see a number of carotenoid genes. So carotenoids produce the bright reds and oranges and yellows that we see in nature. So a number of genes there. Some of these genes have really clear potential roles in flickers. So CYP2J19, for instance, we know from canaries, this is actually one of the only genes we know right now that changes yellow carotenoids into red carotenoids. We know this from canaries. And we find this related to the wing and the tail color in flickers, which makes a lot of sense because this is a yellow versus red coloration difference. So we have some clear associations, but we also have some interesting patterns here. So we have a lot of melanin genes, but these melanin genes are actually mostly related to carotenoid traits. So things like the malar stripe, the bright red patch, or the malar stripe on the face, the nuchal patch on the nape of the neck, this bright red patch on the nape, and the color of the wing and the tail, which is really interesting biologically because melanins and carotenoids are not produced or processed in the same way. Melanins can be produced within the body, carotenoids have to be eaten and then can be processed. But the way that all of this processing and deposition of these pigments happens are completely different. So the fact that we're finding melanin genes associated with carotenoid traits is kind of confusing. And we don't know why this is yet. This is one of the things I'm actively working on in my lab here. But I'll just say feathers are complicated structures. So going from the actual pigment compound out to the color that we see, there's a ton of steps in between. And genetics are likely involved in many of these. And one of the things that recently I've gotten really interested in, sort of leveraging for my science, is aberrant sightings of birds. So aberrant flickers may provide some clues to what's going on here. So here's some interesting sightings of aberrant flickers. The one in the middle is actually the one that got me really interested in this. This was a sighting in Massachusetts in 2015, which was early on in my PhD when I had started working on flickers, but had not, done all of this work I'm telling you about today. So let's just zoom in on this individual. So this is a yellow-shafted individual we know from the region it was found in. We also know you can see it's just flush with yellow. So underneath these feathers that would usually be brown, there's yellow carotenoids throughout the body. We can see it has the tan face, the tan ear coverts, tan throat, as we would expect. Hard to see what color the crown is here. The other thing we can see, so let me show you what, again, yellow-shafted and red-shafted flickers would typically look like. The other thing you might notice is that the malar stripe here, which I told you is always black in yellow-shafted flickers, is red. So this is a male individual that has a red malar stripe, but is yellow-shafted. Now, it turns out what's happening in these malar stripes of yellow-shafted flickers is they have a red patch that they just put melanin on top. So they take red, they add a bunch of black pigment, and it becomes a black patch. So perhaps what's happening with this relationship between melanin genes and carotenoid traits is the color of the plumage in these birds is not as much related to what's happening with carotenoid genes, but it's what's happening with the melanins. Where are the melanins being deposited? And that gives us the final color that we see. So this is something we're actively interested in. I love sightings of aberrant birds, and yes, I could talk about them forever. But that's something we're interested in and pursuing, trying to understand what's going on with this relationship here. Okay, so thinking about the genetic basis of color, what's going on in flickers? So I've told you about some work that we've been doing here, and we've identified a number of differentiated regions between these birds and their genomes. By and large, these birds are really genetically similar, but there are some regions that are differentiated, and these tend to associate with color, such that we have pigment genes within them, and some of them with very clear potential roles in flickers. But we're working on increased ability to sequence these birds and hoping to understand more about what's going on in the story soon. So what I've told you about today is a bit about plumage coloration. I told you a little bit about how to identify these birds. I've told you some about the hybrid zone and how I'm using these same identification of color skills to look at how the hybrid zone has moved. So over the past 60 years, we know the hybrid zone has moved about 73 kilometers, sort of west towards the Rocky Mountains. And then I've also told you a little bit about the genetic basis of color in flickers, about how these birds differ just generally in their genomes. And we find few differentiated regions in the genome, and the ones that we do find are by and large associated with coloration differences. Okay, so I'll just put this slide up here, thanks to lots of organizations and people that have helped fund this work. And I'll be happy to take any questions. I'll close this so I can see you all.
Thank you, Stepfanie. That was really wonderful talk. Yeah, let's open it up to the floor for